System Biosciences has been a leader in the field of extracellular vesicles biology with our simple yet innovative tools for EV isolation, analysis, engineering, and quantitation cited in over 6,000 peer-reviewed publications. One of our widely used families of products is SmartSEC. It isolates EVs from human plasma and serum, and it alone has been cited in more than 50 publications. It's the fuel for your innovation. Today, we're excited to introduce a new addition to the SmartSEC family, a breakthrough in extracellular vesicle isolation. It's called SmartSEC TC, and it's designed specifically for use with tissue culture media. If you're working with tissue culture media, this kit will be your go-to solution for faster, simpler, and ultra-pure extracellular vesicle, or EV, isolation. The SmartSEC TC kit builds on our proven SmartSEC technology, combining the precision of size exclusion chromatography with advanced contaminant trapping technology. This ensures exceptional purity, high yield, and reproducibility that you simply won't find in conventional SEC or resin-based isolation methods, all from just a single fraction elution, while preserving the integrity of your EVs. And that's not all. It's ideal for a wide variety of downstream applications, including Western blot, nanoparticle tracking analysis, transmission electron microscopy, next-gen RNA-seq, and proteomic and lipidomic profiling. The full-size kit is divided into 2 boxes. The first box contains 8 media concentrators, while the second box includes 8 columns, a bottle of column solution, and 8 column closures. Each media concentrator contains a membrane component that retains the EVs after concentration via centrifugation. The concentrated volume can be estimated using the markings on the side of the concentrator. SmartSEC TC streamlines your workflow. Here's how it works. First, transfer the tissue culture media sample into a 15 ml Falcon tube. Place the Falcon tube and a balancing tube into the tabletop centrifuge, then close the lid. Centrifuge at 3,000 times G for 15 minutes to pellet the cellular debris. Remove the Falcon tube from the tabletop centrifuge. Carefully transfer 10 ml of the pre-cleared media into the filter portion of the media concentrator. The media solution is now ready for concentration. Now, place the media concentrator and a balancing tube into the tabletop centrifuge and close the lid. Centrifuge at 3,000 times G for 5 to 30 minutes until the volume is reduced to 300 to 600 microliters. Check the solution volume every 5 to 10 minutes. If the volume is still too high, place the media concentrator back into the centrifuge until it is reduced to 300 to 600 microliters. Sample volume has now been reduced to 600 microliters or less and is ready for the next step in the protocol. Carefully transfer the final concentrated sample into an Eppendorf tube. Multiple transfers may be required if using a 200 microliter pipette tip. Place the tube containing the concentrated media in an ice bucket. Centrifuge to remove any debris. The concentrated media sample is now ready for EV isolation. Twist off the bottom closure from the SmartSEC TC column. Place the column into an empty 15 ml conical collection tube and loosen the column cap. Place the column in a tabletop centrifuge with a balance tube and centrifuge at 500 times G for 1 minute. Remove the column from the centrifuge. Add 1 ml of column buffer to the column to wash it. Return the column to the tabletop centrifuge and centrifuge at 500 times G for 1 minute. Repeat this step once more for a total of 2 washes. Place a closure on the bottom of the SmartSEC TC column. Gently transfer the concentrated media sample onto the top of the pre-washed SmartSEC TC column. Avoid transferring the cell degree pellet at the bottom of the tube. Secure the column by replacing the cap. Gently invert the tube 5 times. Place the SmartSEC TC column containing the EV sample on a rotator for gentle mixing for 10 minutes. Remove the SmartSEC TC column containing the EV sample from the rotator and place the SmartSEC TC column containing the EV sample into a new 15 ml Falcon tube for EV collection. Remove the bottom closure and loosen the cap halfway. Place the column containing the EV sample into a tabletop centrifuge. Centrifuge at 500 times G for 1 minute. Remove the Falcon tube from the centrifuge. Your EV sample will be collected at the bottom of the tube. Carefully transfer the EV sample to a new tube. The entire process takes less than 1 hour and no more than 10 minutes of hands-on time. Your EVs are now ready for a wide range of downstream applications like Western blotting, NTA, TEM, RNA sequencing, proteomic and lipidomic profiling, and more. EVGuard, a non-penetrating cryoprotectant, maintains the biological activity of your sample during long-term storage and multiple freeze-thaw cycles. EVs isolated from cell culture media using SmartSEC TC possess typical EV morphology: intact vesicles with lipid bilayer membranes with little visible background debris, as shown by transmission electron microscopy here. SmartSEC TC isolated EVs show definitive signals of EV protein markers as analyzed by the ExoCheck exosome antibody array. So why should you use SmartSEC TC? Quite simply, SmartSEC TC is faster, easier, and cleaner than traditional methods, making EV isolation from tissue culture media effortless. With a simple reagent-free workflow, you'll get high yield and purity, quick and efficient results, and a seamless experience for consistent, reliable outcomes. Best of all, it only requires elution of a single fraction, so there's no need to collect multiple fractions, no need for a fraction collector, and no need to routinely re-optimize. Welcome to the world of SmartSEC TC, the fuel for your innovation.